Welcome to the deep dive. Today we're looking at the science, the really critical science behind what many call the biggest public health threat we face. We're diving into how bacteria manage to change, to adapt, really, with incredible speed.
And we're starting with, well, a pretty striking image to get a sense of the scale involved. Our sources point out how the environment really pushes this evolution. Think about agricultural waste, for example. Just one dairy cow, a high-producing one, can make about 150 pounds of manure every single day.
Wow, 150 pounds. And if you multiply that, across all the livestock globally, that's not just waste. It's this huge, warm, nutrient-packed incubator.
Exactly. It's a massive reservoir for bacteria carrying antibiotic resistance genes, or AR genes.
And there's a clear link, right? The livestock often get antibiotics.
Yes, sometimes for treating infections, but historically also at low levels in feed to promote growth. This creates, well, intense selective pressure on all the bacteria living in and around them. Adapt or die, essentially.
And the really worrying part, as the research shows, isn't just that resistant bacteria pass these genes down to their offspring, their daughters' cells. It's how they share them. Sideways, you could say.
That's the crux of it. Sharing genes horizontally across different individuals, even different species. That's what makes microbial evolution so incredibly fast and frankly, dangerous.
It drives this crisis that leads to, over 30,000 deaths each year, just in Europe and the U.S., from infections that antibiotics just can't touch anymore.
That's the grim statistic, yes.
So our mission in this deep dive is to unpack how they do it. How do these tiny organisms, which don't reproduce sexually like we do, manage to swap these powerful survival tools so effectively? We'll explore the three main strategies: mutations, their sophisticated DNA repair systems, and the big one for rapid change, horizontal gene transfer, or HGT.
Right. Let's start at the beginning, the ultimate source of new genetic traits, mutations. Simply put, these are stable changes in the DNA sequence that can be passed down. They're the sort of raw material for evolution to work with.
And we're not necessarily talking huge changes all the time.
No, not at all. Most are what we call point mutations. Just one single DNA base pair is affected. It might be swapped for another, or one base might get inserted or deleted. There are larger mutations too, insertions, duplications, but they're generally less frequent.
And where do these spontaneous changes come from? Inside the cell.
Mostly internal processes, yeah. One big source is errors during DNA replication. The copying enzyme, DNA polymerase, isn't perfect. Sometimes the DNA bases themselves briefly shift into a slightly different chemical form, a tautomer.
A tautomer.
Yeah, like a temporary chemical wobble. It changes how the base pairs up just for an instant, but it can trick the polymerase into inserting the wrong base opposite it. That leads to a substitution, a transition, or a transversion in the new DNA strand.
So the base's own chemistry flickers, and boom, a potential mutation. It really shows how delicate the whole process is.
It does. Another internal source is spontaneous damage, like losing a base entirely, leaving a gap, an AP site.
And that's all before we even consider outside threats, things causing mutations.
Right, induced mutations from mutagens in the environment. We tend to group these based on how they damage the DNA. Some chemicals, base analogs, look like normal bases but pair up wrong. Others actually modify the bases chemically.
And then there are ones that physically disrupt the DNA.
Exactly. Intercalating agents are flat molecules that slide between the base pairs, like sticking a card into a deck. This distortion often causes insertions or deletions during replication, leading to frameshift mutations, which are usually really bad news for the resulting protein.
The classic mutagen example always seems to be UV light.
Ah, yes, UV radiation. It causes a very specific kind of damage, forming bonds between adjacent thymine bases, thymine dimers. These dimers create a big kink in the DNA strand that blocks replication machinery.
So once a mutation happens, what does it actually do to the protein the gene codes for?
Good question. We classify them by their effect. A silent mutation changes the DNA codon, but thanks to redundancy in the genetic code, it still codes for the same amino acid. So no change to the protein.
Okay, harmless then.
Usually. Then there's missense. This swaps one amino acid for another. The effect here can vary wildly. Maybe it does nothing, maybe it slightly alters function, maybe it completely kills the protein's activity.
And the really harmful ones.
Those would be nonsense and frameshift mutations. A nonsense mutation changes a normal codon into a stop signal. Translation just stops short, making a truncated, usually useless
And
protein.
frame shifts, you mentioned those with intercalating agents.
Right. If you insert or delete one or two bases, not multiples of three, it shifts the whole reading frame downstream. Every codon after the mutation is read incorrectly, leading to a completely garbled and almost always non-functional protein. Very disruptive.
Okay, so we've got mutations happening spontaneously and being induced.
Yeah.
If you're in the lab, how do you find the bacteria that have these specific mutations?
There are different approaches, is
Okay.
right? Yes, selection versus screening. Selection is generally much easier if you can do it. You set up conditions where only the mutant you're looking for can grow. For example, plating bacteria on a medium with an antibiotic. Only resistant mutants survive. The wild type, non-mutants, die.
So you select for the trait you want. And screening.
Screening means you have to check every colony, usually hundreds or thousands, to find the one with the different phenotype you're interested in. Think about looking for an auxotroph, a mutant that's lost the ability to make something essential, like an amino acid. You might use replica plating to transfer colonies to different media to see which ones fail to grow without that nutrient. It's much more labor-intensive.
That technique, looking for mutants, is really central to the Ames test, isn't it?
Yeah.
For safety testing.
Absolutely. The Ames test is a clever application. It uses strains of Salmonella bacteria that are already mutants. They can't make the amino acid histidine, so they're his auxotrophs. The core idea is that many things that cause cancer, carcinogens, are also mutagens.
They damage DNA.
Exactly. So you expose these his bacteria to the chemical you're testing. If the chemical is a mutagen, it might cause reversion mutations, meaning it mutates the already mutated histidine gene back to a functional state.
So the bacteria suddenly can make histidine again.
Right. And if they can make histidine, they can grow on a medium that lacks it. So if you see lots of colonies growing where there shouldn't be any, it signals that the chemical is mutagenic and therefore potentially carcinogenic. It's a really important initial screening tool.
Okay, so errors happen constantly from inside and out. Cells must have ways to fix this damage, otherwise genomes would just fall apart. This brings us to DNA repair.
They absolutely do. And these repair systems are crucial for stability. You need some variation from mutations, but too much is lethal. These repair mechanisms are remarkably conserved across all domains of life. You even see extreme examples like Deinococcus radiodurans.
Ah, the radiation-resistant bug.
That's the one. It can survive incredible doses of radiation that would shatter most genomes. Interestingly, its secret isn't necessarily having unique repair enzymes, but more about how its genome is structured and how resistant its proteins are to damage, allowing it to piece its DNA back together efficiently.
So what are the main repair strategies, the everyday ones?
Well, the very first line of defense is proofreading. This happens right during replication. DNA polymerase itself has a kind of backspace key, a 3 to 5 foot exonuclease activity. If it adds the wrong base, it often pauses, removes it, and tries again.
Okay, immediate correction. But what if it misses one?
That's where mismatch repair comes in. This system scans newly replicated DNA for errors that proofreading missed, and it has a really clever trick to know which strand is the original template and which is the new, possibly incorrect strand.
How does it do that? That seems vital. You don't want to fix the correct template strand.
It uses methylation. For a short time after replication, the original parental DNA strand is methylated at specific sites, like GATC sequences in E. coli, while the newly synthesized strand is not yet methylated, it's hemimethylated. The mismatch repair proteins recognize this difference, bind to the mismatch, and specifically cut out the error from the unmethylated new strand. Then DNA polymerase fills the gap correctly.
Wow, a temporary chemical flag to ensure they edit the right copy.
Yeah.
That's elegant.
It really is. Then you have systems for fixing actual damaged bases or distorted DNA, like those thymine dimers from UV light. Excision repair is key here.
Excision meaning cutting things out.
Exactly. Nucleotide excision repair, or NER, handles bulky lesions that distort the helix. An enzyme complex like Uvrate ABC in E. coli recognizes the distortion, cuts the DNA backbone on both sides of the damage, removes that whole chunk, and then polymerase and ligase fill and seal the gap.
Okay, cut and patch for big problems.
Pretty much. There's also base excision repair, BER. This targets specific damaged bases, maybe chemically altered ones. A specific enzyme called a DNA glycosylase recognizes and flicks out just the damaged base, leaving the backbone intact but creating an AP site, a spot missing its base. Other enzymes then come in, cut the backbone at the AP site, remove that sugar phosphate, and then patch it up.
So more like targeted single base removal. Are there ways to fix damage without cutting anything out?
Yes. Direct repair mechanisms. Photoreactivation is a classic example. An enzyme called photolase uses energy from visible light to directly break the bonds, forming a thymine dimer, restoring the original bases. No cutting needed. Another is alkyltransferase enzymes that directly remove harmful alkyl groups like methyl groups that might have been added to bases.
Okay, lots of specific tools. But what happens if the damage is just overwhelming? Like the replication fork completely stalls?
That triggers the emergency system, the SOS response. This is really a last resort for the cell.
The life or death switch you called it earlier.
Pretty much. When DNA damage is so severe that it physically stops DNA replication, single-stranded DNA regions accumulate. A protein called RECA binds to this damaged single-stranded DNA. This binding activates REGA's second function. It becomes a coprotease.
The coprotease.
It helps another protein, the Lexa repressor, destroy itself. Normally, Lexa keeps a whole suite of DNA repair genes switched off, but when RECA is activated by damage, it triggers Lexa to cleave itself, lifting that repression.
Okay, so suddenly dozens of repair genes get turned on. That sounds like a good thing.
It is, initially. Genes for excision repair, recombinational repair, they all get boosted. It even turns on a gene that temporarily stops cell division, giving the cell time to fix things. But there's a huge downside, a dangerous gamble.
Which is?
If the damage still can't be repaired conventionally, the final part of the SOS response kicks in, translesion DNA synthesis. The cell activates special DNA polymerases, POL4 and POL-V, in E. coli, which are error-prone. They lack proofreading ability
Why would it use error-prone polymerases?
because they can replicate past damaged sites that would normally block the main polymerase. They essentially guess what base should be there or just put in anything to get replication moving again.
So the cell survives, it finishes replicating its DNA.
But at the cost of introducing potentially many new mutations throughout the genome, it prioritizes immediate survival and completing replication over maintaining genetic accuracy. It's a mutagenic repair system, a desperate move.
That desperate mutagenic state sounds like a perfect setup for rapid adaptation, leading us to horizontal gene transfer, HGT. If mutation provides the variation, HGT seems like the way to spread successful variations incredibly fast.
Absolutely. HGT is arguably the main engine driving microbial evolution and adaptation, especially for things like antibiotic resistance. It's gene transfer between independent mature organisms, completely distinct from vertical transfer from parent to offspring. Genes can move between distantly related species this way.
When some donor DNA, the exogenome, gets into a recipient cell, creating what's called a morozoite, what can happen to it? Does it always stick?
No, there are basically four fates. One, it can get integrated into the host chromosome. This usually requires recombination, often homologous recombination. Two, if the donor DNA is a plasmid capable of replicating on its own, it can persist and replicate as an independent element. Three, if it can't replicate and doesn't integrate, it'll just get diluted out and lost as the cell divides. And four, the host cell has defense systems like restriction enzymes or CRISPR-Cas that might recognize the foreign DNA as non-self and simply degrade it.
You mentioned recombination for integration. What are the main ways that happens?
The most common is homologous recombination. This requires the incoming donor DNA to have a significant stretch of sequence similarity or homology to the recipient's DNA. Proteins like RECA then mediate the exchange, essentially swapping the donor segment for the homologous recipient segment.
Okay, needs similarity. What if there isn't much similarity?
That's where site-specific recombination comes in. This doesn't rely on long stretches of homology. Instead, it uses specific enzymes called recombinases integrases, resolvases, transposases that recognize specific short DNA target sequences on both the donor DNA, maybe a virus or a plasmid, and the recipient chromosome. The enzyme makes cuts at these sites and joins the donor DNA into the recipient DNA. It's very precise, used by many viruses and mobile elements.
Okay, let's break down the three big mechanisms of HGT itself. First up, transformation.
Transformation is, conceptually, the simplest. It's the uptake of free, naked DNA directly from the environment. When bacteria die and lyse, their chromosomes break apart, releasing DNA fragments.
But can any bacterium just grab this DNA?
No, the recipient cell needs to be in a specific physiological state called competence. Only competent cells can bind and take up external DNA. This state is often triggered by specific conditions or growth phases, like in Streptococcus pneumonia during early exponential growth.
And how does the DNA get in and integrate?
Typically, the double-stranded DNA binds to the cell surface. An enzyme, an endonuclease, often associated with the uptake machinery, usually degrades one of the strands. The remaining single strand is then actively transported into the cell. Once inside, this single strand can align with its homologous region on the chromosome and be integrated via homologous recombination mediated by RESA.
Okay, naked DNA uptake. Next is conjugation. This one needs contact, right?
Yes, direct cell-to-cell contact. It's often called bacterial mating, although it's a one-way transfer. It requires the donor cell to have a special type of plasmid called a conjugative plasmid, like the famous F factor or F plasmid in E. coli.
F for fertility.
Exactly. A cell with the F plasmid is F plus us. It produces a structure called a sex pilus, which is actually part of a complex secretion system, a type IV secretion system, or T4SS. This pilus makes contact with a recipient cell that lacks the F factor, an F cell.
And then the transfer happens.
The pilus retracts, drawing the cells close. A signal triggers the F factor DNA to be nicked at a specific site, the origin of transfer, or ority. Then, using a process called rolling circle replication, one strand of the F factor DNA is transferred through the T4SS bridge into the F cell. Both cells synthesize the complementary strand, so the donor remains F plus A, and the recipient becomes F plus A.
So in this F plus by F cross, mainly the plasmid gets transferred, not usually the host's own genes.
Correct. Chromosomal gene transfer is rare in a simple F plus X FF mating, but there's a variation that transfers chromosomal genes very efficiently.
Ah, that must be HFR.
That's HFR, high frequency of recombination. This happens when the F factor, which is an episome, meaning it can exist independently or integrate into the chromosome, actually integrates itself into the host bacterial chromosome.
Okay, so the plasmid is now part of the main chromosome.
Right. Now, when this HFR cell initiates conjugation, the T4SS is assembled, and the DNA is nicked at the ority within the integrated F factor. But because it's integrated, what starts transferring first is part of the F factor, followed sequentially by the host chromosome genes adjacent to it.
So it drags the chromosome along with it.
Precisely. The connection between the cells is usually fragile and breaks before the entire chromosome and the rest of the F factor can be transferred. The result is that the recipient cell efficiently receives a chunk of the donor's chromosomal DNA, which can then recombine, but it usually doesn't receive the full F factor, so it remains F.
Clever. So HFR allows efficient transfer of chromosomal genes. Is there another variant involving the F factor?
Yes, F prime, or F conjugation. This happens when an integrated F factor in an HFR cell excises itself imprecisely from the chromosome. It loops out incorrectly and takes a piece of the adjacent host chromosomal DNA with it.
So it becomes a plasmid again, but carrying extra bacterial genes.
Exactly. This new plasmid is called an F factor. When this F cell conjugates with an F recipient, it transfers the F plasmid, including those captured bacterial genes. The recipient becomes F and is now partially diploid. It has its own copy of those genes plus the copy that came in on the F plasmid.
Okay, transformation conjugation. The third HGT mechanism is transduction. This involves viruses, right?
Yes, bacteriophages or phages viruses that infect bacteria. Transduction is virus-mediated gene transfer. There are two main types.
What's the first one?
Generalized transduction. This typically happens during the apoptotic cycle of a virulent phage, the cycle where the virus replicates rapidly and destroys the host cell. During the assembly stage, when new phage particles are being put together, the packaging machinery sometimes makes a mistake.
How so?
Instead of packaging the viral DNA into the new phage head, it accidentally packages a random fragment of the host bacterial DNA. The fragment size is usually about the same as a phage genome size.
So you get a phage particle carrying bacterial DNA instead of viral DNA.
Exactly. This is called a transducing particle. It looks like a normal phage. It can still attach to and inject its DNA into a new bacterial cell, but it's injecting bacterial genes from the previous host, not viral genes. It's non-infectious in terms of causing lysis. And because the initial packaging was random, any part of the bacterial chromosome could potentially be transferred this way.
Okay, that's generalized. What about the other type?
That's specialized transduction. This involves temperate phages that can undergo a lysogenic cycle, where their DNA integrates into the host chromosome as a prophage and remains dormant.
Like the F factor integrating.
Similar idea, yes. Specialized transduction occurs when this prophage is induced to exit the chromosome and enter the gelitic cycle, perhaps due to host cell stress. If the excision process is sloppy, if it loops out incorrectly, it can pick up a small, specific piece of bacterial DNA that is located immediately adjacent to the phage's integration site on the chromosome.
So it's not random, like generalized transduction.
No, it's specific to the genes flanking the prophage integration site. The classic example is phage lambda in E. coli, which integrates near the GAL, galactose metabolism, and biosynthesis genes. When lambda excises improperly, it can create transducing particles carrying gal or biogenes.
Okay, that covers the main transfer routes. But what about the genes themselves, especially resistance genes? How do they move around so easily, getting onto plasmids or into phages? This sounds like transposable elements.
Precisely. Mobile genetic elements, or transposable elements, often called jumping genes, are key players. They are segments of DNA that can move from one location in the genome to another, or even between a chromosome and a plasmid via a process called transposition.
What forms do they take?
The simplest are insertion sequences, or IS elements. They're quite small and typically only contain the gene coding for the enzyme that allows them to move, called transposase, flanked by short, inverted repeat sequences that the transposase recognizes.
So they just carry the machinery for jumping?
Yes. But then you have transposons, which are more complex. They contain the transposase gene plus additional genes very often, antibiotic resistance genes. Composite transposons have extra genes sandwiched between two IS elements. Unit transposons carry their resistance genes and their own transposition enzymes within a single unit defined by inverted repeats.
And how do they actually move? Do they just jump out and land somewhere else?
There are two main mechanisms. Simple transposition, sometimes called cut and paste, is where the transposase cuts the element out of its original location and inserts it into a new target site. The original site is left empty or repaired.
Okay, cut and paste. What's the other way?
Replicative transposition. In this mechanism, the transposon is copied, and the copy is inserted at a new target site, while the original transposon remains in its initial location, so the number of copies increases. This is particularly significant for spreading genes like antibiotic resistance.
Right, because you're duplicating the resistance gene every time it moves.
Exactly, and this brings us right back to the antibiotic resistance crisis. These AR genes are very often found located on mobile elements like transposons, or on plasmids, our plasmids resistance plasmid, that are themselves often studded with transposons, or within larger mobile chunks of DNA called integrative conjugative elements, ICEs, or mobile genomic islands.
So resistance genes are packaged onto these mobile platforms.
Yes. And because these platforms, the transposons, the plasmids, the ICEs, can move within genomes and between cells via HGT mechanisms like conjugation and transduction, they allow for the incredibly rapid dissemination of multiple resistance genes at once. A bacterium can become resistant to several different antibiotics simultaneously by acquiring just one R plasmid or mobile island carrying multiple resistance transposons.
And it connects back to the very beginning, the selective pressure. Even low levels are enough.
That's a critical point from the research. You don't need full therapeutic doses of antibiotics to drive this. Even very low sublethal concentrations, sometimes as little as 1% of the concentration needed to kill the bacteria, the MIC, or exposure to other stressors like heavy metals found in agricultural runoff, can be enough selective pressure to favor the maintenance and spread of these resistance genes within bacterial populations in the environment.
It's really an incredible picture of adaptability we've painted here. From tiny chemical flickers causing point mutations to complex repair systems like the risky SOS response, all the way to this vast HGD network sharing entire gene cassettes across species boundaries. Bacteria just seem relentless in finding ways to evolve.
They really are, and the speed is the key challenge for us. This constant rapid evolution fueled by mutation, sometimes error-prone repair like SOS and especially HGT, allows microbes to consistently stay one step ahead of our interventions, like the development of new antibiotics.
So thinking about that SOS response again, that high-risk, high-reward survival strategy where the cell basically says, forget accuracy, just replicate, makes you wonder.
What are you thinking?
Well, with the constant low-level presence of antibiotics and other stressors in our environment, in the water, in the soil, stemming from forces like that manure we started with, are we inadvertently pushing vast populations of environmental bacteria towards using that mutagenic SOS response more often? What's the hidden evolutionary cost, or maybe the hidden danger, of constantly nudging bacteria into this error-prone, hypermutagenic state? Could it be accelerating the emergence of completely novel resistance mechanisms that could then jump into pathogens?
That's a deeply unsettling and important question. What are the long-term consequences of pushing microbial evolution into overdrive in this way? We don't fully know, and it underscores the complexity of the challenge we face.
A truly fascinating, if sobering, deep dive. Thank you for walking us through these intricate microbial mechanisms today.